Good morning. It is my pleasure to introduce our speaker for today's lecture, Dr. Hashimoto, who will give us a lecture titled Translational Research for Intracranial Aneurysms. Dr. Hashimoto is a professor of neuroanesthesiology and neurobiology and director of the Translational Neurovascular Research in the Barrow Aneurysm and AVM Research Center. He is board certified in anesthesiology by the American Board of Anesthesiology and works at the Barrow Neurological Institute. Dr. Hashimoto's expertise include clinical anesthesiology and vascular biology. He earned his medical degree from Gifu University School of Medicine in Japan. He received his residency training in anesthesia at Gifu University Hospital, Thomas Jefferson University Hospital in Philadelphia, and the New York Presbyterian and Columbia University Medical Center. He completed a clinical fellowship at the New York Press and Columbia Medical Center and a clinical research fellowship at the University of California in San Francisco, both in neuroanesthesia. These are some of the selected publications where you can see all the work and the research that Dr. Hashimoto leads. And he leads a program that focuses on translational research of cerebrovascular diseases. His group studies the role of inflammation and vascular remodeling in the pathophysiology of intracranial aneurysms and brain AVMs. They are developing pharmacological therapies for the prevention of stroke caused by rupture of intracranial aneurysms and brain AVMs. In representation of the Mayo Clinic Neurosurgery Department, we will be sending this plaque all the way to Arizona to thank you for your time and your lecture this morning, Dr. Hashimoto. Welcome.
Also, Dr. Hashimoto, before you start, we would like to acknowledge the great attendance that we had this morning. We have 30 attendants in person and 60 virtual participants from 9 different countries, including Argentina, Colombia, Dominican Republic, Spain, Mexico, Italy, Lebanon, and multiple states in the US. So welcome and thank you for being here.
All right, thank you so much for a nice introduction. You guys have such an exciting research program, and my research talk may not be as exciting as your research program. But today I'm going to talk about translational research of intracranial aneurysm. I have no conflict of interest. So my research group is interested in pharmacological prevention of aneurysm rupture. So for unruptured aneurysm, clipping and endovascular procedures are used for the prevention of aneurysm rupture. So clipping and coiling are the methods for primary prevention of aneurysm rupture. However, the adverse outcome rates after clipping or coiling of unruptured aneurysm are not quite negligible. If you look at international study of unruptured intracranial aneurysm, the adverse outcome rates, significant mortality and significant morbidity combined maybe as high as 10 to 20%. So for the high volume centers like yours may have much better outcomes, but when you think about the nationwide or worldwide, the adverse outcome rates for clipping or endovascular procedures for unruptured aneurysm are not quite negligible yet. And these therapies are technically intensive and costly. Cost may be an important issue as we are detecting more unruptured aneurysm. So more unruptured aneurysm are requiring treatment. And there may be a limited treatment option for patients with large complex aneurysm. So we think pharmacological stabilization of aneurysm for the prevention of rupture may be an attractive alternative approach. So this diagram shows the disease course of aneurysm, formation and growth of aneurysm, and probably many of the aneurysm become stabilized at some point, and they do not rupture. However, some of the aneurysm become destabilized. They keep growing. These aneurysms may rupture. So the idea of pharmacological prevention of aneurysm rupture is by providing short-term medical treatment, we want to change the natural course from destabilization to stabilization so that we can prevent aneurysm rupture. So to understand why some of the aneurysm rupture and to develop medical therapies for pharmacological prevention of aneurysm rupture, we thought it'd be useful to have a mouse model of intracranial aneurysm. But when we started this project, there was no animal model in which aneurysm rupture happens. There were only animal models for aneurysmal formation, but not so for aneurysm rupture. So we wanted to create a mouse model that produced large aneurysm at a high instance within a short incubation period. And because we wanted to study the mechanism for aneurysm rupture, and we wanted to develop medical therapies for the prevention of aneurysm rupture, we wanted to make sure in our model, aneurysm do rupture. So to create a new mouse model of intracranial aneurysm, we focused on two factors that are associated with human intracranial aneurysm. The first one is high blood pressure, hypertension. That is because hypertension is associated with intracranial aneurysm formation rupture. And hypertension is considered as a risk factor for aneurysmal rupture. And hemodynamic stresses triggered by hypertension is considered as an important role for the formation of aneurysm. And for the second factor, we focused on the disruption of elastic lamina. That is because the disruption of elastic lamina is a histological hallmark of human intracranial aneurysm. And the disruption of elastic lamina alone has been considered as a pre-aneurysmal change that lead to aneurysm formation. So our simplistic idea was we want to combine chronic hypertension and the disruption of elastic lamina, and then we can create aneurysm formations. So for our initial studies, we used 8 to 12-week-old mice. And for chronic hypertension, we used DOCA sort hypertension. DOCA is an aldosterone analog. So DOCA salt hypertension combines administration of DOCA, high salt diet, and unilateral nephrectomy. This method has been widely used to study the end organ damages caused by hypertension. This is a classical method for inducing chronic hypertension in many species. And for the disruption of the elastic lamina, we used a single injection of elastase into the CSF. We tested many different chemicals and enzymes for the disruption of elastic lamina, but we found that elastase injection is the most effective way to cause the disruption of elastic lamina. So for the injection of elastic lamina, we use stereotactic approach as shown in the picture on the left. And we inject the elastase into the right basal system as shown in the picture on the right. Blue dye shows the spread of elastase after a single injection of elastase. And with this method, elastase activity lasts only 2 to 6 hours in the CSF. So this is a short-term treatment with elastase. So when we combine chronic hypertension and a single injection of elastase into the CSF, we were able to induce aneurysm formations in mice. These pictures shows representative aneurysm from our model. Picture on the left shows an unruptured aneurysm in the posterior circulation. And picture in the middle shows another unruptured aneurysm in the anterior circulation. And picture on the right, this is a picture from a mouse that developed motor deficits 11 days after aneurysm induction. When we euthanized this mouse, when this mouse developed motor deficits, we found subarachnoid hemorrhage. And within the hemorrhage, we saw an aneurysm formation So this was a probably ruptured aneurysm. So in this model, 60 to 80% of mice developed aneurysm within 3 to 4 weeks. And from a histological standpoint, these aneurysms closely resemble human intracranial aneurysm. And when we look at leukocyte infiltration into the aneurysm wall, just like a human intracranial aneurysm, most of the leukocytes in the aneurysm wall was macrophages. So a combination of a chronic hypertension and a single injection of elastase into the CSF resulted in intracranial aneurysm formations in mice. And from morphological and histological standpoints, these aneurysms closely resembled human intracranial aneurysm. So initially, using this model, we studied a mechanism for the formation of aneurysm. We found that macrophages are critical for the formation of aneurysm, at least in this model. And we found that matrix metalloproteinases from macrophages, such as MMP9 and also macrophage PPAR gamma, are required for the formation of aneurysm. And my group and other groups that have used our model have shown the key roles of inflammation and inflammatory cells in the formation of aneurysm. But more importantly, these studies have established the feasibility of using this model to study mechanism for the formation of aneurysm. So I showed you the same diagram before: formation of aneurysm, growth, stabilization, and destabilized aneurysm rupture. So the pharmacological stabilization is to change the natural course from destabilization to stabilization. And many of the earlier studies that use our model or other models have focused on the mechanism for the formation of aneurysm. Those earlier studies were based on the assumption that by studying the mechanism for aneurysm formation, we would understand more about the mechanism for aneurysm rupture. However, this assumption may not be correct when you consider a long time interval between the formation of aneurysm and rupture of aneurysm. So mechanism for aneurysm rupture may be fundamentally different from mechanism for aneurysm formation and growth. So we thought, can we directly study the mechanism for aneurysm rupture using this model? So as I told you before, when we are doing our initial studies, we found that some of the mice suddenly developed motor deficits. And when we euthanized these symptomatic mice, we found aneurysmal subarachnoid hemorrhage. So we thought maybe we can use neurological symptoms to detect aneurysm rupture in this model. So in the next study, we looked at the time course of motor deficits or neurological symptoms a little more carefully in a larger sample size. So this graph shows a symptom-free survival curve, symptom-free survival on y-axis, x-axis time. So day 0 is a time when we induced aneurysm. So as you can see, for the first 7 days, none of the mice developed motor deficits. But after 7 days, some of the mice started developing motor deficits. And when we systematically euthanized these symptomatic mice, all of them had ruptured aneurysm and subarachnoid hemorrhage, showing that neurological symptoms, motor deficits, can be used to detect aneurysm rupture. And we verify this method of detecting aneurysm rupture by using daily MR imaging. So this is a time course of our model. So aneurysm formation happens during the first 7 days after aneurysm induction, and aneurysm rupture develops during the second and third week, and rupture rate is about 70 to 80%. So our idea was, if we start medical treatment, if we start a drug treatment at day 7 after aneurysm induction, so if we start a drug treatment after aneurysm already formed, can we test a drug treatment for the prevention of aneurysm rupture? And using the same paradigm, can we study mechanism for aneurysm rupture? So as a first test case to see whether we can use this model to study the mechanism for aneurysm rupture, we focused on hypertension. Again, hypertension is a risk factor for aneurysm rupture. However, a causal link between systemic hypertension and the development of aneurysm rupture has not been established. Well, that is because at the time of diagnosis of unruptured aneurysm, the majority of patients are already treated with antihypertensive agents. So we cannot test causal relationship between hypertension and aneurysm rupture in humans. But we may be able to do this in mice. So the question we asked was, does hypertension directly contribute to the development of aneurysm rupture? So this is a question we asked in our mouse model. Does the normalization of blood pressure reduce aneurysm rupture? So again, aneurysm induced by a combination of DOCA-sort hypertension and a single injection of elastase. And for the normalization of blood pressure in this model, we use a direct vasodilator, hydralazine. So this graph shows our systolic blood pressure on y-axis, x-axis time. So day 0 is the time when we induce aneurysm. And you can see a gradual increase in the blood pressure, and blood pressure stayed high. And then we started the treatment with the antihypertensive agent 7 days after aneurysm induction. So we studied the antihypertensive agent after aneurysm formed. And you can see hydralazine effectively reduced blood pressure, normalized blood pressure. Then what happened to the rupture rate? So this is a rupture rate. Y-axis indicate rupture rates. X-axis shows vehicle group and hydralazine group. So the normalization of blood pressure by hydralazine significantly reduced aneurysm rupture, indicating a causal relationship between hypertension and aneurysm rupture. So we extended this study using other antihypertensive agent, ACE inhibitor, angiotensin type 1 receptor antagonist. And as far as the mechanism goes, our studies are indicating the activation of local renin-angiotensin system in the vascular wall or aneurysmal wall is responsible for the hypertension-induced aneurysm rupture. Hypertension activates the local renin-angiotensin system, and the activation of local renin-angiotensin system leads to vascular inflammation and abnormal remodeling, leading to aneurysm rupture. So we are able to show a causal link between hypertension and aneurysm rupture using this model. So this study established the feasibility of using this model to study mechanism for the development of aneurysm rupture. And we are able to establish the feasibility of using this model to test pharmacological therapies for the prevention of aneurysm rupture. So after this study, our group has been focusing on the prevention of aneurysm rupture. So one of our current projects focuses on aging and aneurysm rupture. That is because epidemiological studies have shown the age-dependent increase in the incidence of subarachnoid hemorrhage. So this graph is from a systematic review by DeLuige that studied 17 epidemiological studies This graph shows in both men and women, there's a clear increase, age-dependent increase in the incidence of aneurysmal rupture. So what is a biological basis for the age-dependent increase of aneurysmal rupture? And if we identify the mechanism, can we target aging-related biology for the prevention of aneurysm rupture? So to start this, before we start this project, we wanted to make sure our model recapitulates age-dependent increase of aneurysm rupture. So for this study, we compared 12-week-old mice and 18-month-old mice. 12-week-old mice corresponds to young adults in human, and 18-month-old mice correspond to 55 to 65 years old in human. Those are the median ages for aneurysm rupture in human. So these are the results from male mice. Graph on the left shows rupture rate. As you can see, aged mice had significantly higher rupture rate, just like a human. And we confirmed this by plotting symptom-free survival curve. And we saw the same effect of aging on aneurysm rupture in female mice as well. So our model was able to recapitulate age-dependent increase of aneurysm rupture that was observed in humans. So in terms of mechanism to study, we are interested in aging and senescence. There are many biological changes that are associated with aging, but senescence is a hallmark of aging. Senescence is a state of permanent proliferative arrest. After cells divide so many times as we age, these cells stop dividing, but they don't die. They become senescent cells. So senescent cells cannot divide, but they don't die. They are the dysfunctional cells that accumulate in our body as we age. And they secrete pro-inflammatory cytokines and tissue remodeling cytokines. These cytokines are collectively called senescence-associated secretory phenotype, SASP. So SASP factors can damage tissues and promote local inflammation, causing many of the phenotypes of the aging. But there are two types of senescence in our body. The most one is aging-related senescence. As we age, cells divide, but they stop dividing. But cellular stresses induced by hypertension, shear stress, smoking, and other factors can cause premature senescence. So in our body, as we age, we are looking at increased senescence burden that come from combination of aging-related senescence and premature senescence. And these senescent cells secrete SASP factors. They can cause inflammation, abnormal remodeling, and wall damage. And SASP factors themselves can cause senescence, making a vicious cycle. And this vicious cycle may lead to aneurysm rupture. So that is our hypothesis. So the first question we asked using our model is, do senescent cells promote aneurysm rupture? So for this, we used this transgenic mouse, P16-3MR mice. This is a really unique mice. These are really unique mice. So in P16-3MR mice, they have a DNA construct that has P16 promoter, and at the downstream of P16 promoter, they have two cell markers, luciferase and modified red fluorescence protein. And more importantly, at the downstream of P16 promoter, these mice have HSV-TK, herpes simplex virus 1 thymidine kinase. So P16 is a senescence marker. So when the cells undergo senescence, P16 promoter gets activated. So when P16 promoter activated, these transgenic mice start producing two cell markers and HSV-TK. HSV-TK is a herpes simplex virus 1 thymidine kinase. This thymidine kinase should exist only in herpes virus. So when we give ganciclovir to the patients with herpes, ganciclovir attacks thymidine kinase inside the herpes, that causes damage to the viral DNA, and that kills the herpes virus. But in this particular transgenic mouse line, when the cells are undergoing senescence, when P16 promoter is activated, these senescent cells, only senescent cells, express HSV-TK. So if we give ganciclovir to these mice, we can selectively kill, we can selectively eliminate senescent cells. So this tool was developed by Drs. Campisi and Di Maria. So this mouse line has almost nothing to do with a human disease, but this is an extremely useful tool to study the laws of senescence in many disease processes. So in P16 3-MR mice, we induced aneurysm, then we gave ganciclovir so that we can selectively eliminate senescent cells. So this is a result. Graph on the left shows incidence of aneurysm. There was no difference in the overall incidence of aneurysm between two groups. So the elimination of senescent cells did not affect the incidence of aneurysm. And graph on the right shows the rupture rate. As you can see, ganciclovir treatment, elimination of senescent cells reduced rupture rate. This is not statistically significant yet. This is a preliminary data. We are trying to increase the sample size to confirm this trend. And we saw the same effects of elimination of senescent cells in male mice. Elimination of senescent cells did not affect the overall incidence of aneurysm. So the elimination of senescent cells did not affect the formation of aneurysm, but it did reduce the rupture rate, indicating that senescent cells may promote aneurysm rupture. So the next question is, can we use medications? Can we use medical therapies to attack senescent cells for the prevention of aneurysm rupture? So there are class of drugs called senolytics. Senolytics are drugs that reduce senescent cells. Senolytics selectively attack senescent cells. There are many senolytics. Some of them are already in clinical trial. So we chose to use ABT263 as our senolytics. So first we wanted to make sure when we give senolytics to mice, we are able to reduce senescent cells. So we measured expression of P16 in cerebral arteries of the mice. So P16 is a senescence marker, and ABT263 treatment effectively reduced P16 expression, indicating that the treatment with these senolytics can reduce senescent cells. And then this is the incidence of aneurysm. Just like the case of P16 3-MR mice, senolytics did not affect the formation of aneurysm. However, senolytics significantly reduce rupture rate, indicating that we can use senolytics to reduce senescent cells for the prevention of aneurysm rupture. So we're still working on this project. We still need to connect many dots so that we can explain why there is an age-dependent increase in the incidence of aneurysm rupture, and so that we can understand how we can target aging-related biology for the prevention of aneurysm rupture. And there are two types of drugs we may be able to test. Senolytics are drugs that can selectively eliminate senescent cells, and xenomorphs is an exciting drug they can suppress SASP factor production. So we may be able to use these drugs for the prevention of aneurysm rupture. So today, I talked about the development of mouse model of intracranial aneurysm in our model, in our lab. And we showed our effort to establish the feasibility of using this model to study mechanism for the aneurysm formation and then mechanism for the aneurysm rupture. And I talked about one of the current projects we are working in our lab. And this lists some of the past and current projects. Many of them are focusing on the mechanism for aneurysm rupture. And I want to thank my collaborators, including Dr. Lawton and Dr. Eguchi from Temple University. And the support for my research comes from Barrow Neurological Foundation and Brain Aneurysm Foundation and the National Institute of Health. Thank you.
Wonderful presentation. I'm Rabi Talk, one of the faculty here. Amazing talk, basically looking at all these mechanisms, and the way you finished it was kind of the most intriguing to me, that there is so many ways to focus on and there is so many pathways. If you have to select one of those, which one you think is the highest yield? Because we all know that aneurysm, and the conclusion also, what you came up with, the aging and the blood pressure are the highest two risk factors. So how would you integrate all those and work on one thing if you have to change? Because we know it's multifactorial, but which one would you go with? Which one would be your top choice to work on?
Yeah, I think that's a very good point. We can, at least in the mouse model, we can prevent aneurysm rupture by attacking different pathways, but many of them kind of focus on the inflammation. So attacking inflammation may be one of the best strategy, because if you think about all the risk factors like smoking, hypertension, aging, they are all linked to inflammation. So probably attacking inflammation in one way or another would be the most promising choice. But in terms of the choice of drugs, we still don't know. We would like to do a comparative study of different methods.
Great. And the second question is related to markers. We can select blood samples from a vein, which is peripheral blood sample, or you can go inside the aneurysm or inside the vasculature, or tandem to the aneurysm, where the aneurysm kind of release things and you go distal. Do you think there is a role for markers for work like this?
Yeah, I think there is a great value for looking for biomarkers, and we are working on the biomarkers by analyzing blood samples and human aneurysm specimens. But one of the difficulty is, as you know, if you look at the risk factors again, like smoking and high blood pressure, and maybe extremely high has a high odds ratio. So when we try to look for the biomarkers, we may end up finding biomarkers that are associated with high blood pressure or smoking. So, but those biomarkers do exist in patients without aneurysm as well. So I think it's really promising, but it might be quite difficult to find the biomarkers that are truly unique to aneurysm formation or aneurysm rupture.
You just shut down my third question, which was which biomarker you select, but you answered it already. So thank you so much. Amazing work.
Thank you so much. Great talk, very interesting and thought-provoking. I'm Chris Fox. I'm one of the other vascular guys here working with Dr. Talk. I had a similar question to what he was going to ask, but since he asked it, I have another one. You know, we do a lot of 7T MRI here, and it also makes me think about, you know, our vessel wall imaging. And I'm just curious if you guys are looking at that, and if you've seen any changes, you know, in either, you know, patients, and that guides your clinical approach, or with your model, if you've been able to do that, looking at a radiographic biomarker.
Yeah, I agree. The vascular wall imaging is one of the most exciting area in the imaging of aneurysm. But we are not doing any active project at this point because as far as mouse aneurysm goes, they're too small for even 7 Tesla MRI. And for human studies, at least to me, many of the studies may not be kind of objectively assessed. So when I look at, I'm not the imaging specialist, I don't have any training in imaging, but when I look at their representative pictures, even in the paper, so they must be the best pictures. But I cannot quite see true difference between, let's say, unruptured aneurysm or ruptured aneurysm, or what they call like unstable versus stable aneurysm. I think probably that there will be there is a difference probably we can detect, but I think it's really difficult at this point to study as a biomarker. I think they need to develop much better technology to objectively assess the vascular wall intensity.
Yeah, I agree. I sometimes have to really squint to look at the differences between, stable and unstable vessel wall imaging. But I'll just put in a plug since, we're doing so much 7T here, and Dr. Middlebrooks is very active with our group. And we also have a huge biobank that is, run by Dr. Q, as you know. So, maybe some potential for collaboration going forward.
Yeah, I think combining tissue analysis with actual vascular wall imaging and correlating the finding between them would be really important.
And I was just going to echo, this whole presentation, Dr. Hashimoto. I think that both Dr. Talk and Dr. Fox illustrate the power of thinking of multi-institutional projects, grants, biobanking, all the way from blood, which is what they do every day when they go in, to tissue potentially that surgery needs to be done, to biomarkers, all the way from imaging. I think that the last plugin to consider is how do we use artificial intelligence for those images that Dr. Fox was mentioning with the 7 Tesla, right? And how do you then create over time in the same patient, the analysis of whatever biomarkers may be on the wall, potentially wall thickness. Does it change too drastically, too fast, not fast enough? How do you predict which patients may require intervention sooner rather than later? And which ones, you can potentially put coils and they're going to do well? Because I think that what I'm seeing when I see the cases that both Dr. Talk and Dr. Fox are doing is that sometimes you have coils and these patients, the wall continues to thin. And then they become problem. You've seen it there at the Barrow with Dr. Lawton as well. These cases become a lot more complex after that. So I think the other opportunity, as I keep telling Chris and Robbie here, and they're interested in collaborating with you, is biologicals. How do we interfere with stem cells, for instance, from adipose tissue, from bone marrow, which I think that you're already working for one of the lines that you put right there. So incorporating that into biodegradable, coils or things that are the next generation. So something to consider if you're interested in collaborating with the team. So thank you.
Yeah.
So I'm going to ask our fellows. So what's the schedule today for Dr. Hashimoto? Because I know that he's going to meet with the residents first in a couple of minutes.
Absolutely, sir. Dr. Hashimoto is going to meet with the residents from 8:05 to 8:30, then from 8:30 to 9 with the neurosurgery consultants, 9 to 9:30 with the research fellows, and 9:30 to 9:45 with Dr. Q. Thank you very much, Dr. Hashimoto.
Thank
Dr.
you.
Hashimoto, we thank you immensely. You can see so many people here in the room. So I sent you some pictures, so many people from around the world joining us. And we thank you for dedicating your morning to us. It means a lot to us. I know there's a few hours difference between you and us right here. Next time, we'd love to have you in person here with us.
Thank you.
Thank you.